Hi everyone, my name is Josselyn and I'm a current graduate student in Dr. Sherry Carnes' lab here at Stanford. Our lab studies transplant immunology and my project specifically focuses on investigating the role of natural killer cells in the control of latent Epstein-Barr virus infection, which is what I'm going to be talking to you all about today. To start, Epstein-Barr virus or EBV is a herpes virus that infects roughly 90% of the global population. EBV infections are mainly asymptomatic. However, failure to control latent EBV infection can result in a variety of malignancies, including lymphoproliferative diseases in immunocompromised people. Studies in both experimental models and humans suggest that NK cells are critical in the host defense against EBV. And so in previous work done in the lab, we demonstrated that NK cells that express the inhibitory receptor NKG2A were specifically able to recognize and kill autologous B cell lines that were latently infected with EBV. We also demonstrated that HLA-E-presented peptides derived from EBV latent glycoproteins can impair NKG2A recognition and its downstream inhibitory signaling, which potentially can lead to NK cell activation. The main question we are still trying to answer is, what is this mechanism behind NK cell recognition of latent EBV? To better understand the phenotype of these NK cells that respond to EBV-infected B cells, we generated a panel of EBV lymphoblastoid cell lines, or EBV LCL, and performed co-culture experiments with these cell lines. As you can see in our first schematic, we drew fresh blood from a given healthy donor and isolated their NK cells using magnetic separation. These NK cells were then co-cultured with either the corresponding autologous EBV LCL, autologous primary B cell, or cultured alone overnight with IL-2 supplemented media. Co-cultured NK cells were then sorted for CD107A via FACS and sequenced via 10x genomics single-cell RNA sequencing. The subsequent analyses that I'm showing you were completed in R using the SIRAT-V3 package and are from 2 healthy donors. So my second figure here shows our UMAP separation between samples. And so UMAPs of the sequencing data show in part A, clustering based on 8 principal components. And in part B, we can see the distinction between responsive and non-responsive NK cells across all samples, with responsive NK cells being the CD107A positive population in red and non-responsive NK cells corresponding to the CD107A negative population in purple. While there are CD107A positive NK cells throughout the UMAP, LCL responsive NK cells overlap the most with clusters 4 through 6, which are found in the right half of the top circle shape. So with this in mind, we looked further into the relative expression of select genes to visualize the expression levels in certain clusters. In our third figure, the relative expression of several different genes is indicated in red. The genes in part A are genes that appear to be consistently upregulated within our own analyses, such as interferon gamma. The genes that were chosen in part B were markers that were previously found in other studies from our lab to constitute the predominant NK cell population that responds to autologous EBV LCL. And so upregulation of interferon gamma and IL-2RA is specific to the LCL responsive NK cells, as you can see by, again, looking at the right half of the top circle shape that I mentioned previously. And the cytokine CCL3 is upregulated by all responsive CD107A positive cells, with peak expression being found in the same cluster that corresponds to the LCL responsive NK cells. The markers that were chosen in part B, as I mentioned before, correspond to a previously characterized subset of NK cells that respond best to autologous EBV LCL. My sequencing data is able to recapitulate this population that we saw in the lab, as we see expression of NKG2A, NKG2D, and 2B4, and a general lack of NKG2C and CD57 in our own LCL responsive NK cells. And so our main conclusions so far are that NKG2A is upregulated in LCL responsive NK cells, and interferon gamma and IL-2RA upregulation also seems to distinguish LCL responsive NK cells from other CD107A positive cells. ISG15 and CCL3 are upregulated by LCL responsive NK cells. ISG15 is an interferon stimulated gene that has been implicated as a central player in the host antiviral response, and CCL3 is important in inflammatory responses to the receptors it binds to. This is one of the first studies to implicate these genes in their response to EBV, and so, of course, we're very excited to see what additional information we can gain about these mechanisms. These analyses, again, show only a small selection of genes, and so, of course, further analysis is still required to better understand the prevalence of additional NK cell markers, as well as other chemokines and cytokine receptors within LCL responsive NK cells compared to non-responsive cells. And right now, I'm also working on additional experiments using a novel mass cytometry panel of over 40 NK functional and phenotypic markers and I'm currently analyzing mass cytometry data for 6 healthy donors in order to validate our sequencing findings and, of course, better understand the phenotype of LCR responses and K cells. And so, with all of this being said, I thank you all so much for the opportunity to present for the Holman Day. I really appreciate your time and welcome any questions or insights that you may have. Please feel free to reach out at the email below. And again, thank you.